students. I am Palak Agarwal and today we will study about PCR. So PCR basically stands for polymerase chain reaction. It is a technique to create multiple copies of DNA or the required gene sequence which was invented by Carey Mullis. This process takes place in 3 steps: denaturation, annealing and extension. We will go into the details of this. Polymerase chain reaction. The principle of this process. What's the principle? Principle is enzymatic replication of DNA. The process. As I told you, process has 3 steps. First step, denaturation of DNA. That is breaking of DNA strands. As you all know, DNA is double-stranded in nature. So, and bonded by hydrogen bonds. Now what happens when we heat the DNA at 90 to 95 degrees Celsius of temperature? The strands of DNA get separated and the bonds between them is broken. These single strands lead us to the second step of the process of PCR, that is annealing of primers. Annealing of primers. Now before we get on to the second step, we need to understand what are primers. Primers are basically 15 to 25 base pairs nucleotides combination called oligonucleotides. The length cannot be more than 25 or less than 15, as the length will create the hindrance of binding of primers to the DNA strands. So now these single strands of DNA, these two single strands both having polarity 5' to 3'. These primers comes and bind to the 3' end. So this is the process of annealing. Annealing means binding. Binding of primers to single strands of DNA or template strands of DNA. Now what happens? There comes an enzyme called DNA polymerase, which leads us to step 3. That is called extension. Now before we study the third step, that is extension, we need to understand the work of this DNA polymerase. So this DNA polymerase is basically Taq polymerase. That means it is thermostable species derived from Thermus aquaticus. This species Thermus aquaticus produces an enzyme called Taq polymerase, which we use for this process, as this whole process takes place in vitro, which is in lab. So after binding of Taq polymerase, what happens? The DNTPs, nucleotides that are present around the template strands, comes and start binding as per the order given by Taq polymerase, and DNA strand starts extending. See what happens? Here it's the primer, here is DNA polymerase. So now one by one nucleotides come and start binding to each other and make a strand in this direction, 5' to 3'. Remember, primer comes and binds to 3' site only as they are complementary to it. And on the other strand that has this polarity, primers will bind DNA polymerase and the extension will take place in this direction. And above this, the extension is taking place in this direction. And in the results we get 2 strands of new DNA and we get 2 new DNAs. Now when these 3 steps are repeated again and again, we get multiple copies of DNA. So after 30 times repeating of these 3 steps, we get 1 billion copies of DNA. After 30 seconds. Now what these 3 steps takes place at particular temperature. Like I told you, denaturation is at 90 to 95 degrees Celsius. For annealing it's 50 to 70 degrees Celsius, and for extension it's 72 degrees Celsius. It might range from 70 to 75 degrees Celsius. And as we talk about applications, the applications include, it can be used for paternity testing to know who the parent of child is, to solve paternity issues cases, DNA testing that we call, and in forensics to check about mutation. So these are some applications of PCR. Thank you.